Okay, thank you. Good morning, everyone. Today, Batul and I have a research presentation on autophagy's role in brain health. So, what is autophagy? Autophagy is a Greek word for self eating, which is a highly conserved pathway and unique mechanism that degrades large organelles and protein via lysosomes. It is the body's way of cleaning out damaged cells in order to regenerate newer, healthier cells. Autophagy is primarily considered a pro-survival machinery. Under physiological conditions, cells perform low levels of autophagy to maintain homeostasis, termed basal autophagy. However, autophagy activity can be upregulated by a variety of life-threatening events such as hypoxia, nutrient depletion, exposure to reactive oxygen species, invasion of microorganisms, organelle damage, and excessive accumulation of aggregated proteins. Now, coming to abnormal autophagy. When the cellular process of autophagy is either excessively activated or insufficiently carried out, leading to a disruption in the balance of cellular component degradation and recycling, it leads to abnormal autophagy, which further contributes to various diseases such as neurodegenerative disorders, cancer, and metabolic conditions. Now, coming to the next slide, that is the neurodegenerative disorders. Neurodegenerative disorders are conditions that gradually damage and destroy parts of one's nervous system, especially areas of one's brain. These conditions usually develop slowly, and the effects and symptoms tend to appear later in life. It represents a group of progressive conditions encompassing a wide spectrum of clinical presentation, including cognitive impairment, motor dysfunction, and psychiatric symptoms. Examples include Alzheimer's disease, Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, and frontotemporal dementia, among others. While these disorders have distinct clinical features and pathological hallmarks, they still share a common underlying mechanism involving protein misfolding, aggregation, and neuronal loss. Now, coming to the link between autophagy and neurodegenerative disorders. It has been seen that abnormalities in autophagy contribute to the development and progression of Alzheimer's and Parkinson's diseases in the following ways. In Alzheimer's disease, impaired autophagy leads to the accumulation of amyloid beta plaques and how protein tangles within neurons. These pathological aggregates disrupt neuronal function, leading to a synaptic dysfunction, neuronal loss, and cognitive decline. In Parkinson's disease, it has been seen that defective autophagy mechanisms fail to clear misfolded alpha synuclein protein aggregates. These aggregates accumulate within neurons, forming Lewy bodies which impair neuronal function and contribute to the characteristic motor symptoms of the Parkinson's disease. Now we have some research studies listed over here. Hamano et al. 2021 in a research study stated that dysfunction of autophagy, which disrupt the effective clearance of misfolded proteins and cytoplasmic oligomers, has observed in many neurodegenerative disorders, including Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, and Huntington's disease. Therefore, regulation of autophagy may be favorable in prevention and therapeutics of Alzheimer's disease by preventing how protein aggregation. Another study done by Lynch Day et al. 2012 stated that there have been tremendous advances in the understanding of the causes of Parkinson's disease. Novel genes causing familiar Parkinson's disease have been discovered and have been shown to be involved in the autophagy pathway, one of the major proteolytic symptoms that maintains cellular protein homeostasis. It was further stated that discovery of therapeutic agents that boost autophagic activity or that directly maintain the mitochondrial homeostasis could potentially reduce neuronal loss and slow down the disease progression. Now over to you, Batul. She will be taking the link between autophagy and mental disorders.
Dysfunctional autophagy can significantly impact how the body responds to various forms of stress, leading to detrimental effects on cellular and overall health. Now we will take a detailed look at how look at the consequences of dysfunctional autophagy in the context of stress. So first we have psychological stress, under which we have chronic stress. So, dysregulated autophagy can exacerbate the effects of chronic psychological stress by failing to manage the cellular damage and inflammation it causes. This is associated with the development and progression of mental health disorders, including depression, anxiety, and stress related cognitive decline. Next, we have stress hormones. So, dysfunctional autophagy can lead to an abnormal response to stress hormones like cortisol. This can result in a maladaptive stress response, further contributing to mental health issues and impairing the body's ability to cope with stress. Next, we have cellular stress response, under which we have oxidative stress. So, when autophagy is impaired, damaged mitochondria and other organelles are not efficiently removed, leading to the accumulation of reactive oxygen species. This can result in increased oxidative damage to cellular components, contributing to aging, cancer, and other diseases. Next, we have nutrient deprivation. A dysfunctional autophagy impairs the cell's ability to recycle components to generate energy and essential nutrients during periods of starvation. This can lead to cellular energy deficits, decreased survival under nutrient stress, and impaired function of vital organs. Lastly, we have hypoxia. Impaired autophagy hinders the removal of damaged proteins and organelles, which is critical under low oxygen conditions. Cells may fail to adapt to hypoxia, leading to cell death and contributing to diseases such as ischemic injury. Now we'll move on to the next disorder. Can we go to the next slide, please, Solanky? Thank you. Now we will look at the link between autophagy and depression. Dysfunctional autophagy can have a profound impact on depression, influencing the onset, progression, and severity of the condition. Now we will look at now we will take a detailed look at how impaired autophagy contributes to depression. So firstly, there is neuroinflammation. Dysfunctional autophagy can lead to the accumulation of damaged cells and proteins, triggering chronic inflammation. Increased inflammation is a well-documented contributor to depression. Pro-inflammatory cytokines can alter brain function, disrupt neurotransmitter systems, and affect mood regulation. Next, we have neurotransmitter imbalance. So, impaired autophagy can disrupt the balance of neurotransmitters such as serotonin, dopamine, and norepinephrine. Imbalances in these neurotransmitters are closely linked to the development and persistence of depressive symptoms. For instance, reduced serotonin levels are a hallmark of many depressive disorders. Next, we have mitochondrial dysfunction. Dysfunctional autophagy leads to the accumulation of dysfunctional mitochondria, resulting in reduced cellular energy production and increased oxidative stress. Mitochondrial dysfunction has been associated with fatigue, cognitive impairment, and mood disturbances, all of which are common symptoms of depression. Next, we have synaptic plasticity. So, impaired autophagy can lead to the accumulation of damaged synaptic components and reduced synaptic remodeling. Impaired synaptic plasticity is associated with cognitive deficits and mood disorders, including depression. This can affect an individual's ability to adapt to new experiences and stressors. Next, we have brain-derived neurotropic factor. Dysfunctional autophagy can lead to reduced levels of brain-derived neurotrophic factor, and lower levels of this are linked to the pathophysiology of depression. This deficiency can result in neuronal atrophy and reduce neurogenesis, contributing to depressive symptoms. And lastly, we have stress response. So, when autophagy is impaired, cells are less capable of managing stress effectively, leading to increased vulnerability to stress induced damage. Chronic stress is a major factor for depression. Ineffective stress response mechanisms due to dysfunctional autophagy can exacerbate the impact of stress on mental health, increasing the risk of developing depression. Now we'll move on to the last disorder. Can we go to the next slide, please, Solanky? Yeah. Now we look at the link between autophagy and schizophrenia. Autophagy is in emerging research suggests that dysfunctional autophagy may be implicated in the pathophysiology of schizophrenia. And now we look at how this works. So, firstly, we have neurodevelopmental factors. Schizophrenia is thought to involve neurodevelopmental abnormalities. Impaired autophagy during critical periods of brain development may contribute to these abnormalities, increasing the risk of developing schizophrenia. Next, we have synaptic function and plasticity. Synaptic dysfunction is a core feature of schizophrenia. Impaired autophagy can exacerbate synaptic deficits, contributing to the cognitive and behavioral symptoms of the disorder. Next, we have neuroinflammation. So, increased neuroinflammation is observed in schizophrenia. Persistent inflammation can disrupt brain function and contribute to the pathophysiology of the disorder. Next, we have neurotransmitter systems. Schizophrenia is associated with imbalances in neurotransmitter systems, particularly dopaminergic hyperactivity and glutamatergic dysfunction. Dysregulated autophagy may contribute to these imbalances. Next, we have genetic links. So, understanding the genetic links between autophagy and schizophrenia can provide insights into the molecular mechanisms underlying the disorder and identifying potential targets for therapeutic intervention. And lastly, we have oxidative stress. So, elevated oxidative stress is a feature of schizophrenia. Impaired autophagy can exacerbate the oxidative stress, contributing to neuronal damage and the progression of the disorder. Now we look at the conclusion. Abnormal autophagy has far-reaching effects on stress responses, mental health disorders like depression and schizophrenia, and neurodegenerative diseases. By disrupting cellular homeostasis, synaptic function, and neurotransmitter balance, and mitochondrial health, this functional autophagy exacerbates the pathophysiology of these conditions. Understanding the specific roles of autophagy in these disorders offers promising avenues for therapeutic interventions. Modulating autophagy through pharmacological agents or genetic approaches holds potential for mitigating the effects of these disorders, improving patient outcomes and quality of life. Solanky will take up the quiz now.
Thank you, Batul. So, the first question is, what is autophagy? A, it is the body's way of cleaning out damaged cells in order to regenerate newer, healthier cells. B, it is a digestive function. And C, it is a cardiovascular function. Yes, A is the correct answer. Thank you for participating. Second question is, abnormalities in autophagy contribute to the development and progression of which neurodegenerative diseases? A, Parkinson's disease, B, Alzheimer's disease, and C, both A and B. Yes, C is the correct answer. And last question, abnormal autophagy impacts neuroinflammation, which increases the likelihood of developing schizophrenia. A, true, B, false. Yes, A is the correct answer. Thank you everyone for participating in this quiz.